In response to inflammatory signals, T cells and other leukocytes move into tissues by passing through or between the endothelial cells lining blood vessel walls, a process known as transendothelial migration. Although cell motility is generally controlled by Rho GTPases, Anne Ridley from King's College London says that little is known about the contribution of Rho proteins to this particular type of cell migration.
So the only studies that had been done before on Rho GTPases in this process were done with rather non-specific inhibitors of several Rho GTPases, for example, bacterial toxins, or they added inhibitors of downstream kinases for Rho proteins. So because we could now knock down each one individually, we decided that what we really wanted to know was which of the 20 Rho GTPases that are expressed in human cells could contribute to this process, and then to study them in detail. And it turned out that RhoA was the most important if you knocked it down individually of all the ones we looked at.
Ridley's group, led by postdoc Sarah Heasman, found that knocking down RhoA affected both the appearance and behaviour of T cells attempting to migrate through a layer of endothelial cells.
The normal T cell, when it's migrating on the endothelium, has a broad lamellopodium at the front and a little uropod at the back. Whereas when we knock down RhoA, most of the cells actually are very rounded and have two thin protrusions extending from opposite sides of the cell. The protrusions are not lacking any dynamics, but the cells overall can't really move because they're trying to go in two directions. They also can't extend the protrusions between the endothelial junctions to go underneath, whereas a normal T cell will stop over a junction usually and round up and then extend a protrusion between the endothelial cell junctions.
To investigate how RhoA controls transendothelial migration, Ridley and colleagues used a fluorescent reporter to monitor the GTPase's activity in living T cells.
We transfected this biosensor into the T cells, and in collaboration with our next door neighbours actually, Leo Carlin and Tony In, we then imaged the cells on a multiphoton microscope, which allowed us to look quite precisely at where RhoA is normally active during the process of crawling on the top of the endothelium and once they go through. So what was really fascinating about this is that the RhoA is active in multiple places on the plasma membrane as they crawl and then go through. And the activity changes very dynamically. And what we found is that there are pulses of activity at the front of the cell that correlate with movement forward. And we also saw pulses at the back in the uropod, which correlate with contraction and movement of that membrane forward.
In fact, a closer look at the cell's membrane dynamics revealed that RhoA has two functions at the leading edge.
What we also saw was that sometimes RhoA was active when the front decided to change direction and retract in one part. So what we believe is happening is that RhoA is acting with multiple different complexes to mediate all these different functions in the T cell. On the one hand, it would have one complex where it would stimulate protrusion at the front, another complex where it would stimulate retraction at the front, and another complex where it would stimulate retraction at the back.
These complexes would include the different downstream effector proteins of RhoA. During leading edge and uropod retraction, the effector proteins could be the ROCK family kinases, which increase myosin light chain phosphorylation to stimulate actomyosin contractility.
First of all, we looked at where myosin light chain phosphorylation was occurring, and we found it both in the uropod and in discrete areas in the front, which could correlate with areas of retraction. And then we wanted to know what knocking down the ROCks would do. We knocked down ROCK1 and ROCK2, and we could reduce transendothelial migration.
Adding a ROCK inhibitor caused many T cells to form long tails at their rear that couldn't detach and retract from the underlying endothelial cells. But these thin extensions were different to the ones formed by T cells lacking RhoA.
In the RhoA knockdowns, nearly all of the extensions we saw were due to protrusions rather than leaving a tail behind. So we felt that ROCK was contributing quite a lot to the RhoA knockdown phenotype, but it wasn't contributing everything. We feel that RhoA is also contributing something to broadening of the protrusion to make a lamellipodium, because these protrusions on the RhoA knockdown cells are very narrow. In the ROC knockdown cells, we could quite often see broad lamellipodia. It's just the tail couldn't detach.
As well as using different effectors for its protrusive and contractile functions, RhoA also seems to be activated by different guanine nucleotide exchange factors, or GEFs.
We chose to look at GEF-H1 because it had been reported to be important at the front of migrating fibroblasts. However, when we looked at its localizations, we found that it was really specifically localized in the uropod of T cells. And we showed that if you knock down this GEF-H1, then the cell tail gets longer, so that would indicate that you needed it at the back for a contraction of the tail. Whereas we presume that other GEFs, which we haven't identified yet, would be involved in activating RhoA in response to signals that are happening at the front. So we'd really like to find out what the exchange factors are at the front that are involved in protrusion and retraction, which we think would be different ones. We'll probably have to do a screen of the different Rho exchange factors, of which there are quite a lot in the human genome. We'd like to work out which downstream targets of RhoA are involved in the protrusion and retraction events at the front as well. We suspect the retraction events are due to myosin light chain phosphorylation, but we don't absolutely know that, and that would be something that we could look at in the future.
In the meantime, you can read more about the different functions of RhoA at the front and back of migrating T cells in the paper by Heasman et al., published in the August 23rd issue of the Journal of Cell Biology.